Hi, my name is Emily. Together with Rachel, we're going to be talking about our project on improving the carbon dioxide absorption in photosynthesis by using CRISPR gene editing. First of all, I'm going to talk about how I got the idea of the project. So basically, it was just I saw a YouTube video about someone talking about CRISPR, and I thought it was really interesting and amazing, so I decided to do a project on that. And then I thought, how can I apply CRISPR to something that is useful for the society or to solve some problems? Then I thought about one of the biggest issues in the world right now, which is global warming. And that's basically how I started my project.
So, hi guys, I'm Rachel. And my partner Emily just talked about how we got the idea. So, for next parts, I'm going to talk about our project summarize. So, the rapidly worsening global warming needs to be mitigated by decreasing the amount of greenhouse gases, such as carbon dioxide, in the atmosphere. Meanwhile, the unpredictable population growth is demanding for more efficient crop yield. Both of these requirements can be satisfied by improving the photosynthetic rate of plants. Enhancing the performance of rubisco drives particular attention, as rubisco is a crucial enzyme that helps to complete carbon fixation. The process of converting atmospheric carbon dioxide into energy-rich molecules. Besides, rubisco is very slow working and tends to bind with oxygen to cause the energy-wasting process of photorespiration. So, this study aims to seek possible ways of increasing the carbon dioxide absorption of plants and algae through the targets of genes regulating the structure and activities of rubisco using CRISPR-Cas9 technology. If successful, there will potentially be a number of benefits on the aspects of moderating global warming and increasing crop yield. It is hypothesized that through editing and rearranging DNA sequence using CRISPR technology, the carbon dioxide absorbing rate can be increased by around 30%. So for next parts, I'm going to talk about the methods. So I split this into 4 parts. So first, we decided to determine the genes that encode for rubisco. And then we review the reports of past attempts for genetic engineering about rubisco. And then we try to compare those reports, and we chose the one that is most suitable for applying the CRISPR technology. The last, we come up with a way to combine the CRISPR technology with the genetic engineering of rubisco that we describe in the report, so which involves the next 2 parts. So first, we determine the guide RNA which binds with the region of DNA that is aimed to be cut off. Second, we come up with a method to replace the catalytic DNA with a new DNA sequence.
I am going to briefly talk about the results. The detailed version of our result can be found on our poster or in our report. So we reviewed 3 scientific articles on the genetic engineering of rubisco. It is shown in the first article that the carboxylation rate of tobacco can be increased by around 35% with transformed RBCL genes from Flavaria bidentins. Similarly, the second article talked about transgenic rice with RBCS gene from sorghum that demonstrated high catalytic turnover. Carboxylation rate and catalytic turnover are both parameters measuring the speed of rubisco catalytic carboxylation reactions. In the third article, it is shown that the carboxylase activities of tobacco also increased with replaced rubisco from cyanobacteria. However, the increase in the speed of rubisco catalyzed carboxylation activities in all 3 examples mentioned above are based on sacrificing the carbon dioxide specificity. The carbon dioxide specificity is rubisco's tendency to bind with carbon dioxide rather than oxygen, which we talked before that will cause photorespiration. The sacrifice of carbon dioxide specificity somewhat counteracts the increase in carboxylation rate and reduces its effect in improving the performance of photosynthesis overall. In another important article we found, it is proved that there is a negative correlation between carbon dioxide specificity and catalytic turnover. The article also pointed that genetic editing of rubisco cannot significantly increase the performance of photosynthesis overall. So at the end, our hypothesis of editing and rearranging the DNA sequence using CRISPR technology, the carbon dioxide absorbing rate can be increased by around 30%, is rejected. In terms of applying the CRISPR technology, since genetic engineering of rubisco cannot significantly increase the carbon dioxide absorbing rate, it is not necessary to apply CRISPR onto it. And that's basically all of our project. We definitely face a lot of difficulties during the process. For example, I actually got my idea in 2020, but throughout the whole year of 2020 to 2021, I just wrote one paragraph of thing. The scientific journals that I read for background research were just so intimidating for me. They are very long with many words that I cannot understand. And I never really started the project until September 2021. And I still can't not believe that I have actually finished the project. I definitely learned a lot during the experience. I think that for scientific researches, there's going to be a lot of difficulties and obstacles, but you just have to get the process started. It is not as hard as you think when you start a project. And I love the experience. I enjoyed it a lot. And I think BASIF is a great opportunity for me. Thank you.